So this is lab 3. 2 experiments we're doing today. Common aseptic transfers. Right? And so you'll notice on your homework, right, a lot of this is the procedure that we gave to you ahead of time. And then the other method is streak plate method of isolation that we're going to do. You're going to work in pairs, and you're going to watch each other. You're going to be each other's lab assistant. You watch. You do not grab onto anything or anything like that, right? In my last class, one person was holding the tube for the other. I was like, no, that's not how it works. You have to be able to do this independently. And because you guys are beginners too, watching each other, and I'm going to have you guys watch me, and I'm going to purposely try to remember to make a mistake and see if you guys catch me and let me know, hey, you need to do this, right? Catch me before I make a mistake, right, and help me out. And I expect you to do the same for your lab partner, okay? Because I can't watch all of you, right? It's just not possible. I'm one person. So you got to help each other out on this one. So we started kind of doing like a glossary of terms. I'm not going to go over these, but they're here in the PowerPoint for you guys to reference. And I kind of go over them as I lecture, of course, but sometimes it's nice to have it all in one spot, like you guys do have on your homework. So aseptic means that we're not contaminating, right? And that's twofold. We don't want to contaminate the cultures we're working with or ourselves. So you're going to start with pure cultures, the stock cultures that I have up front. And we use 3 different types of media preps or food preps for our organisms, cultivating vessels as well as types of food. So a liquid culture is referred to as a broth culture. A slant is a solid culture that's in a tube, and the tube has been put on a slant so that we have a surface, this slanted surface, to grow our organisms on. As opposed to a petri dish, right, which is also solid agar, solid food source, and lots of surface area. The problem with these is they're difficult to store, and the lids can easily be knocked off the culture. So most of our stock cultures that we'll work with throughout the semester will be on slants. It's very easy to store, takes up a whole lot less space, especially since we have space issues right now. So our sterile media has no organisms, right? Sterile means no organisms. So we're going to work with all 3 different types of media today, so you guys are familiar with all of them. So we're going to do things like moving an organism from a broth to a broth, and from a slant to a slant, and from a plate to a broth. And I think I'm missing one more here. A broth to a slant, probably. So inoculation means we're introducing organisms into sterile media, right? Into a new food stock. Inoculum is the actual organisms we're moving. And you can pick up a lot, and that would be called a heavy inoculum, right? Where you have a large amount of organisms, and hopefully I will teach you not to do that. Sometimes it's necessary for most of what you guys are doing. You need a light inoculum. You just want to pick up a little bit of organisms, because a little bit goes a long way, right? And so that's what you can't really see in my video that hopefully you'll see in the live demonstration today. It amazes me sometimes when I look at stock cultures and I'm like, where did all of those organisms go? They took way too much. So aseptic transfers is also sometimes referred to as sterile technique, right? And again, we're trying not to contaminate things. We're trying to keep it as clean as possible. There are 2 basic types of stock media that we use in the laboratory. One is called nutrient agar, so we commonly abbreviate it NA. The other is nutrient broth, and the difference between an agar and a broth is agar. Agar is a solidifying agent and creates that solid surface, whether it be in a slant for a tube or for a petri dish. For a broth, the agar is omitted because that's a solidifying agent. It's like gelatin, right? Gelatin, unfortunately, though, is a protein. It does not even stay solid at room temperature. Never mind sticking in at 37 degrees at body temperature, around 98 degrees Fahrenheit, right? It would definitely become goo. That, and just like ourselves, bacteria like to eat that protein, right? And so they would eat it and eliminate it anyways. So agar is a polysaccharide from marine algae, and we haven't found any bacteria or microorganisms to date that actually eat it. So that's good. So it stays solid, right? They don't use it as a food source. It's just a solidifying agent. All the rest of what's in the agar is the food that they'll absorb from that source. So, where is it? So beef extract, as the name implies, right, it's basically cow meat, right? And so just like for us, right, this is the fast food diet, right, good old hamburger, basically. Peptone is a digestion, again, of animal protein. So this is a very diverse food source. It's what we refer to as being complex. We don't know exactly what they're eating. It's not all measured out, but that's okay, because we really just want them to grow. We want a rich source of nutrients, and we just want them to grow. So it's not defined. It's just supportive. Trypticase is another one that we commonly use just for growing, just for propagating our organisms, what we call stock culture media. Tryptone is an enzymatic digestion of casein, which is the milk protein. It's actually what makes milk white. It's that presence of that protein. That's why milk has that color. So it's partially digested, just like the animal protein here. And this is like giving them baby food, right? And so they get to skip the step of chewing, right? Because we ground it up for them ahead of time. And this is to make them grow even faster, right? If they don't have that extra step of having to digest it, if it's already partially digested for them, they're going to grow faster. And of course, we grind up food for babies because they don't have any teeth, right? They'd be gumming it to death. And that would really take a long time. Probably wouldn't want to eat. Soybean. So another thing that's a food source here, salt to balance it. And again, notice it has agar in the name, so it does have that ability to be solidified. Right. So usually when we're just growing, we're just propagating, we want to use these rich sources of media like nutrient agar and trypticase soy. Well-defined medias, this is where we know exactly all the ingredients. And we have just a few of those in this lab. That's not why we're using them. We're using them for other purposes. And I always kind of equate this to the Nutrisystem diet, right, where you know exactly what you're eating. It's also very expensive and really not necessary for what we do in lab, right, to have like really defined, we know what they're eating. We're not doing high-tech experiments. We're propagating organisms for you guys to learn the different properties and play around with them. Selective media are usually also are rich sources of media, but then other things have been added to it. And so we've actually already utilized them. We'll utilize a couple of different selective medias. So I have one of our plates that I'll pass around and talk about in a moment from last time that fall into this category. So why do we transfer them? Right? We want to revive them for maybe a stock culture that's been refrigerated or freezed, and we want them actively growing, right, so that we can study them. Usually we're going to use a loop, and so your loops are up front here. In the beaker, it says loops. So it's a metal loop, right, used to pick up the organism and transfer from one place to another. Serological pipettes sometimes are also used, especially when you're dealing with frozen stocks. Differential or selective tests, right? This gives us more information. We're not just growing them. We can answer certain questions. So we can identify certain abilities of the organisms by utilizing these specialized medias. Again, usually we're going to use a loop, but some of them require the use of a needle. The difference is a needle, people can't breathe, doesn't have a loop at the end. It's literally just a metal rod. Differential media, usually a wide range of organisms will grow on it, but they're going to grow differently depending on the abilities of the organism. So for some organisms, we look at fermentation. For two of the selective differential media that we use, it actually has both properties of being selective and differential. For whether they can ferment sugars. this blood agar plate is differential for what we call hemolysis, where it can actually damage the red blood cells. So when you hold this plate up, you can see clearing where they've destroyed the red blood cells. It even has a greenish tinge to this plate, right? So you can tell that destruction of the red blood cells has happened to this plate. So go ahead and pass that around. And that was from somebody's mouth, right? You have these types of organisms in your mouth. In addition, that plate is selective. That plate contains two antibiotics in it that stop the growth of gram-negatives. And so it's gram-positives that are growing on that plate. One of the other plates we use has a high salt concentration. So only organisms that can resist that high salt situation will grow. So with selective media, it usually has something toxic to the organisms we don't want growing and facilitates the growth of the ones we do want growing. So high salt, all the ones that can't handle are going to die off, and the ones that we're looking for, which are the salt-tolerant ones, will be able to grow in that condition. Make sense? All right, so a little bit of foreshadowing to the type of food sources we'll use in the future in this lab. So why is it important to use aseptic technique? Well, we want to make sure we're just studying what we think we're studying, right? We don't want to be studying contaminants or other things that get into our sample. So we want to keep our original pure culture to be just that. And what a pure culture is a single organism, single species. That's it, right? So that we know we're just studying that one, right? And we don't have the complication of other things being present. So we need to make sure that your sterile media does not become contaminated with one or more additional species, right? We want to keep it that one. Then the next step is, of course, keeping ourselves safe and our environment safe. So we don't want to dump the organisms we're working with into our environment, right? So again, that's where things like gloves help protect us, right? If we have a spill or something like that. We want to keep stuff off of our possessions, off of our body. That's why today is a crucial day when it comes to, because we're using the Bunsen burner, pulling back our hair and putting all our precious belongings in a safe place, the closet. And we don't want to contaminate anyone else either. So I'll pick on the biology labs right now. We had a meeting Monday night, and an instructor must have stepped out of the room or whatnot, and a student spilt a toxic substance, and they left it. Nobody cleaned it up, right? So then the next class that came in was exposed to this and didn't even really know, because it was a powdery substance, and it was all over the place, right? And it was a hazardous chemical. It really needed to be cleaned up and cleaned up properly, because nobody reported it. That wasn't done, right? That doesn't happen in this lab, right? We know how important safety is. Paula likes to joke with us. She calls this the creepy crawly lab. I'm like, this is probably the cleanest lab on campus because we clean up our messes. So we know how important it is to do such. So one of the techniques that we use is sometimes you'll start out with a culture that has multiple organisms. But again, we want to tease it out so that we got individual organisms that we can study separately. So one of the common techniques that's utilized for that is the streak plate method of isolation. So this plate, you can see they're using what's called the quadrant streak, where they're using 4 different sections of the plate to spread out and dilute out the organisms. So hopefully you get them all by themselves, and these what we call isolated colonies because they're all by themselves, or hopefully arose from a single cell or a group of cells of the same species. So we now hopefully have generated a pure culture. There's no guarantee here. And there's some clues I'll let you know sometimes depending on if you knew potentially what you started with. The fact that these are all the same color may be that they're all the same organism, but it also could be similar organisms. Did you guys look at your stock cultures today? Most of the bacteria we work with are creamy white. They all look the same. You're not going to know that it's a particular organism unless the tube is labeled. And if someone labels the tube wrong, you're not going to know unless you run additional tests for sure that it is what it says it is, right? So we're really dependent on being accurate in our labeling and how we do our procedures in this lab. So my other plate I have is an example of a successful streak plate with isolated colonies. So go ahead and pass that one around. And you can see that that organism, and I actually started with pure culture, so they're all the same. And I know what organism that is. With this one, you could guesstimate that maybe you're dealing with a different organism. Because you see red, you see white, and you see yellow. It may be the potential of 3 different organisms. The potential. Again, just because they're different colors doesn't necessarily mean it's a different organism. Organisms are affected by different constraints. So for instance, one of the organisms that I prepped for today is one of my favorite ones, because when you grow it at 25 degrees, that cooler room temperature, it produces a red pigment. When you grow it at 37, though, it's white. Looks like all the rest of the bacteria that we mostly work with in the lab. Right? So for that one, really, temperature will affect it. Right? So I could really screw you up by, you're going to streak it out red and then stick it in 37 and they're going to turn white. Right? The new ones that grow aren't going to produce that red pigmentation under those conditions. So color isn't necessarily mean something's different, organism, right? Because they can change under different environmental constraints. So we're going to use a broth as a source, but you could use any culture media as a source, right, for adding them onto your plate. The reason why we use the plate is large surface area, right, for us to be able to spread them out. So a single streak is what we did before, right, especially for like our desktop, right? There wasn't a lot of organisms, so we could rub and take that swab and rub it all over the plate, and we got, pretty good spread out of the organisms. When you're working with a large number of organisms, you're going to want to, when you have a heavy amount, a heavy inoculum, you're going to want to do the streak plate method. So there's several different ways you can do this. We always draw, remember, on the bottom of our Petri dishes because that's where the food is. The lids could be interchanged, right, and really mess you up. So we'll use our Sharpie marker to draw on the bottom. And especially for beginning students, right, it's always advised that you draw yourself like a little plan. All right, so divide up your plate into the different quadrants. You could angle them, right? There's several different ways you can draw it, right? Do what you think will work for you. So we have 4 different sections. And I'll show you guys how I draw it so that I can actually see my numbers. And they're not inverted. So the first step is going to be adding from your original culture, that mixed culture, your organisms into the first quadrant. And the way that you're going to do that is you're going to start at the edge of the plate and you're going to zigzag back and forth to the line that designates that quadrant. It's invisible coloring. You're not actually going to see. Remember, they're microscopic. And then you're going to go to your quadrant 2, and what I do is I start at the top and I zigzag down. And you're going to cross over into this one a few times, picking up organisms. In some protocols will say, oh, only cross over 2 or 3 times. You guys are beginners. I find it's best and you actually achieve picking stuff up because you're probably not going to get the coverage that a scientist has done this for a long time has, right? So I just say sweep back and forth, and that's what I'll do today, the whole section, right? And as you notice, you're going to be picking up from section 1, putting into 2, and actually into part of 3. So that now when you go to do the section 3, you're picking up from 2 and spreading out. The idea is picking up less and less organisms each time, so that hopefully by the time you get to either the 3rd or 4th quadrant, you have individual colonies. Now this one, since it's a large surface area because we want to get the most spreading out, this is where you really only want to enter in a few times, right? So one way you can enter in a couple of times and then just streak the rest of the section. Or there's another method that I'll show you guys as well. In between each one of these steps, so this is the only time you go into your original inoculum, right? Which you could do that with a swab or a loop, but then after that you're always going to use your loop. you have to flame your loop in between each one of these quadrants, because what that does is remove the organisms from that loop. So they're only picking up ones from one section and dragging them to the other. So as I said, you're picking up less and less bacteria as you go. I like to draw my lines relatively straight, right? Like a goalpost, right? It's easier to draw than trying to do those diagonals and getting good separation. And then some people will draw it such that they have just, quadrant 1, and then pulling off of that quadrant 2 and quadrant 3. So you'll see this pattern sometimes. Again, it's just a guide, right, to help you keep track of where you are and where you're going, right, on your plates. You'll notice that my plate had no lines on it at all, right, and it never did. I freehanded that, right? I've been doing this long enough, I don't need guides, right? And notice mine are more of the diagonal, right, and that's more of how you will be when you get to that point, which most of you guys are not going to get to that point unless you go into research and you have to do these kinds of things a lot. So again, you want to fill in that whole section. You want to spread them out as best you can, right? Flame your loop. Go from 1 to 2. Spread it all out. Flame your loop. From 2 to 3. Spread it all out. And then this is the technique that I teach you guys to do, and that is to just draw a line, pick up your loop, draw another line. So again, hopefully you're just picking up very few organisms, right? But you are hopefully picking up ones that you'll get isolation. You can use either method, but you'll notice, right, you guys have your sheets for recording what you do in lab. Make a note as to which you did, right? So that when I ask you, you know. And I'll demonstrate that when I do my procedure today. So Peter Hickman is the other colleague that I work with in putting materials together for this lab. Shared with the technique he did in research, because he would have to do so many isolations that he would actually divide a plate up into 8 sections, and he'd do mini little streak plates in each one of these sections using sterile toothpicks. So he would take from his original culture and he'd draw just one little tiny line, and then he would use the other end of the toothpick and draw another line off of this one, picking up very few organisms. And then he'd use his next toothpick and he would draw another line, and then the other end of his toothpick he would then drag over the rest of the surface. And so he could, on one particular plate, create 8 different isolates on just a single plate, using it very small, right, with a toothpick even. So you'll start with organisms, right, in slant media and broth media. I want you guys to denote things like the texture. Notice here this one looks very dry, right? Sometimes they'll look very shiny. Denote the color, like in this picture they're all white, but one of the ones you're getting today is pigmented, so you want to make a note of that colorization. For the broth tubes, again, any color, where the cloudiness, where the growth is. Notice in these 2 tubes it's confined to the top of the tube, right? Others it tends to maybe settle out. Others it may be all throughout, especially motile organisms that are able to swim, right? They're going to be all throughout the broth culture. So the cultures we're working with today is Staphylococcus epidermis, which is commonly found on your skin. That's going to be in a slant culture. Micrococcus luteus is also going to be on a slant. Bacillus cereus is actually in a broth. And your Klebsiella pneumonia is going to be on the plate that you have, which for some of those I was able to do some good streak plates, others not so much. And then you'll have 2 different broth mixes that we're going to use for the streak plate. And again, of course, depending on which one you do, you should denote it on your plate and in your notes which mix you ended up doing. But each one of them looks differently too. So, you guys need to work on your observation skills, right? So you should have a little bit different. One of the things that we're not going to go into detail, I'm just going to let you know about. And where's that streak plate? Okay. So we're going to pass the streak plate around again, just the streak plate. I want you guys to keep it and pass it around again. Okay, good, thank you. So what I want you to notice is that if you look at it very closely, the organisms create like a nice little mound. And if you look at the individual colonies, they're nice and round, right? But different organisms, so this one's convex, nice little mound. And it's smooth, right? All the edges are smooth, and the whole colony is round. But other organisms will have these different configurations. I don't want you guys memorizing these names. We're not going to do that. But I just want to bring it to your attention that different organisms will have different growth patterns on solid culture media like on this plate. So here's some examples. And this is one of the ones that we're working with today. Staph epi, and this actually on blood agar plate. But notice again, you'll notice it's white, right? If you were to see the colony, it's raised, right? And again, to really see those structures, you need to look at them under a dissecting scope. They're circular. They're smooth. For this one, Klebsiella pneumoniae, one of the ones we'll work with, sometimes it's going to look very shiny and mucoid-like. This one has a very thick capsule, right? So that's why it has that appearance sometimes. And you'll see even its edges sometimes are kind of irregular. Micrococcus luteus, this is the one you're passing around, right? So notice it's yellow, right? It's convex, it's circular, it's smooth. This organism is on blood agar, I believe. Yes, because it's red. They're white, clearly. They're raised, but notice that especially after 48 hours, 2 days, you get this spreading edge, right? And they're irregular, and they're rough looking and even dry looking. Right, so you're going to, as we go through the semester, really pay attention to what these organisms look like. Right? They do have subtle differences. So one of the things that you're going to do today is make note of those. What do these things look like? And I'm going to leave this slide up, but I'm going to end my recording.